Assalamualaikum and very good morning. Ladies and gentlemen, it is with great pleasure and honor that I stand before you today as a keynote speaker to address a topic of paramount importance in agricultural science, the development of the temperate crops under the soil cooling in the tropics. As Associate Professor Dr. Azizi Othman, I am deeply passionate about exploring innovative solutions to this challenge faced by agriculture, particularly in regions characterized by tropical climates. With the world's population steadily increasing day by day, the conversation around food security and agriculture has never been more critical. As our global community grows, so too does the demand for food. Yet, meeting this demand presents a significant challenge, particularly in the face of climate change, resource constraint, and environmental degradation. As our world population keeps growing, ensuring there's enough food for everyone becomes a bigger challenge. That is where agriculture comes in, but traditional method might not be enough. We need new ideas to grow more food sustainably. As we witness the continual increase in global population, the challenge of ensuring food security for all becomes more pressing. This challenge is intricately linked to Sustainable Development Goal SDG 2, Zero Hunger, which aim to eradicate hunger, ensure food security, improve nutrition, and promote sustainable agriculture. However, it is crucial to note that addressing food security and agriculture extend beyond just one goal. In fact, 12 out of the 17 SDGs directly or indirectly related to agriculture. One promising idea is growing cooler climate crops in hot places. Like the tropics, it sounds strange, but it could make a big difference. By cooling down the soil, we can create conditions where this crop thrive. This could mean more food options and better food security for everyone. In addition to the global challenge of ensuring food security, many countries, including Malaysia, face the pressing issue of low food self-sufficiency. Despite concerted efforts, Malaysia has yet to attain self-sufficiency in food supply, which means it relies heavily on imports to meet its food needs. By investing in innovative agricultural practices, such as exploring soil cooling techniques to grow cooler climates in tropical regions, Malaysia can enhance its food production capabilities and reduce its reliance on imports. This is not only contribute to achieving food self-sufficiency, but also promote sustainable agriculture and economic growth. In the next meeting, I will explore how Malaysia can leverage such agricultural innovation to improve food self-sufficiency and contribute to achieving SDG 2 and other relevant goals. Soil temperature plays a critical role in the effectiveness of nitrogen-fixing bacteria. Optimal temperatures are necessary for these microbes to thrive and perform their nitrogen-fixing function efficiently. However, maintaining the right soil temperature can be challenging, especially in regions with extreme temperature variations. Challenges and solutions such as biotic and abiotic stresses which plants face numerous stresses that can affect their growth. Nitrogen-fixing bacteria help mitigate these stresses by enhancing plant resilience. Temperature regulation, where soil cooling systems can help maintain optimal soil temperatures, ensuring that nitrogen-fixing bacteria remain active and effective. However, these systems can be costly and complex to implement. To address the challenge, innovative approaches are being explored to maintain soil temperatures without relying solely on artificial soil cooling systems. One such approach involves isolation nitrogen-fixing microbe from environment where natural cooling occurs, such as deeper soil layers or shaded area. In tropical climates, growing temperate crops is challenging due to higher temperatures that affect crop yield and soil nutrient availability. While previous studies have used soil cooling to address this issue, these methods are energy intensive and not sustainable. However, the potential of nitrogen-fixing microbes to support temperate crop growth in tropical climates without the need for such cooling has not been exploited. This research gap presents an opportunity to develop more sustainable agriculture practices that can help temperature crops thrive in tropical regions. This slide outlines the setup we use to cool soil for temperate crop cultivation in tropical climates. We use a water chiller set to approximately 4 C, which was connected to a chill water storage tank through circulated pipe. Regular water from the storage tank was cooled down by this chill water and then pumped through embedded pipe within soil plot to reduce the soil temperature. The water was then recirculated back to the storage tank to be cooled again, creating a continuous loop. We set up two experimental conditions, one with thermal conditioning using the cooling system, and another one as a control without cooling. The pipes embedded in the soil plot maintains a temperature range of about 18 C to 23 C, providing a controlled environment to assess the effect of soil cooling on temperate crop growth. This slide presents the growth performance of temperate root crops under different soil conditions. In the tropical climate, we studied a temperature average around 25 C, and soil temperature were significantly higher in uncooled soil compared to cool soil. The result shows that the cool soil, which maintained a temperature of around 21.8 C, lead to substantial improvement in crop performance. Specifically, the radish crop grown in cool soil had a much higher weight of taproot and leaf, longer taproot length, more leaf, and larger leaf area compared to those in uncooled soil. These findings are significant because radish, a temperate crop, perform best at soil temperature between 10 C and 18 C. The higher soil temperatures in uncooled conditions likely stunted growth, reduced biomass, and diminished nutritional value as supported by McGill's 2018 research. This emphasizes the importance of soil cooling in tropical climates for optimizing the growth of temperate crops. Soil temperatures significantly affect the nutrient content in agricultural soil. Our study found that cool soil had a higher total nitrogen content compared to uncooled soil. This is likely due to increased decomposition rate of soil organic matter and enhanced microbial activity in cooler conditions. These findings highlight the importance of soil temperature management for optimizing nutrient availability and crop growth. Our research focused on identifying bacteria capable of fixing nitrogen, a crucial nutrient for plant growth, through various tests. We isolated 3 promising bacterial strains C10, C13, and C21, which shows positive results for nitrogen fixation. These strains possess the necessary genes for this process, making them potential candidates for sustainable agriculture applications. Moving to the next slide, our draft genomes sequence of C10, C13, C21 provides a comprehensive understanding of its genetic agriculture. We identified key genes involved in nitrogen fixation. This information is crucial for developing strategies to harness the nitrogen fixing capabilities of our sustainable agriculture. These are examples of genes in each bacteria like A and F, V and F, NIFD, and NIFK. In this slide, we focus on the gene related to nitrogen fixation in Bacillus sp.C.21. Our draft genome analysis identified several nitrogen fixation-related genes with varying levels of identified to known the genes from other species. Notably, the GLNR gene in Bacillus sp.C.21 shows 100% identity to Bacillus subtilis, where it plays a crucial role in regulating nitrogen metabolism by controlling the expression of glutamina synthetase and other related genes as reported by Sher et al. In 2000. Additionally, we identify genes like A and F and V and F, which are part of alternative nitrogenase enzyme complexes used by some bacteria to fix nitrogen under specific conditions, as noted by Hodgkinson et al. In 2014. This finding suggests that Bacillus sp.C.21 has the genetic potential to fix nitrogen through multiple pathways, making it a promising candidate for supporting plant growth in nitrogen-limited soil. To validate our findings, we conducted field trials inoculating lettuce plants with our selected bacteria strains. Agromyces sp.C10 and Bacillus sp.C21 demonstrated significant growth enhancement compared to the control groups, suggesting their effectiveness in promoting plant growth under tropical conditions. As you all can see in the table, Agromyces sp.C10 and Bacillus sp.C21 significantly increased plant weight, height, leaf number, and root length compared to the control, while Gordonia sp.C13 shows limited impact. Bacillus sp.C21 exhibited the most promising results, particularly when combined with fertilizer. These findings highlight the potential of this bacterial strain for sustainable agriculture and improved crop yield. The research found that specific bacterial strains, which are Agromyces sp.C10 and Bacillus sp.C21, significantly increase plant nitrogen content without the need for soil cooling systems. As reported by Film 2010, greater nitrogen content concentration will have a better growth rate. This suggests a promising approach to reduce fertilizer dependency, improve crop yields, and enhance soil health. Essentially, this highlights the potential of using bacteria as a sustainable solution to boost crop nutrition and productivity. In conclusion, developing temperate crops in tropical climates hold immense potential for global food security. Let's collaborate to unlock this opportunity. Thank you for your time and attention.